I'm Betsy Rothermel. I'm the program director for herpetology and restoration ecology, and it's my pleasure to welcome Dr. Tiffany Doan as our seminar speaker today. She's a new associate research biologist in the herpetology and restoration ecology program here. She's also a research associate and instructor in the Department of Biology at the University of Central Florida. She's a herpetologist and field ecologist who's worked on a wide variety of questions related to ecology and evolutionary biology of lizards, snakes, frogs, wide range of projects. In addition to the project that she's going to tell you about today, she is currently working on projects on global diversity and distribution of reptiles, and continuing research on the ecology of squamates in Peru and Bolivia, where she has worked for, I think, a couple of decades now, at least. So she's going to be talking about the effects of malaria infection on the Anoles of Florida. And hopefully there will also be a little time for her to fill you in on a big research project that is just getting going at the Avon Park Air Force Range. Please welcome Tiffany, and thank you for coming over.
All right, thanks for having me. It's great to meet many of you in person that I hadn't met before. Hopefully I'll get to meet some more of you after the talk. So I currently am working on sort of 3 big projects that don't really seem like they're related to each other at all. But they are related because they're all really looking at phenomena that create biogeographic processes. So I'm really into biogeography, where species are, why are they there, how have humans messed that up? So those are the kind of things that unite these things. So I'm going to spend just a tiny bit of time talking about the other 2 projects and then spend most of the time talking about the malaria project. So global species distributions, disease ecology, and effects of multiple stressors on species are the sort of the 3 big things that I'm doing these days. So I'm just going to spend a minute talking about the global assessment of reptile distributions. So it's a working group that we started in 2008. I'm one of the founding members, and why this happened was because for all the rest of the terrestrial vertebrates, so birds, mammals, amphibians, you could go to a website or open up a book and say, oh, I wonder what species are in this area. And you look in the book and there it tells you. But that's not the case for reptiles. That's still not the case for reptiles. So reptiles, yes, in the US, we have field guides. You can open them up, see what reptiles are here. You can do that in Europe, Australia, Central America. But the rest of the world, no, you still can't do that because there aren't field guides for most of the places. We just don't even know the distributions of reptiles in most of the world. So we got together a team for the areas that were the least well known. So Africa, Southeast Asia, and South America were the ones that we started with. And there were 19 of us when we started our first meeting in 2008 in London. And then since then, we've added a lot more people to our team to sort of broaden the expertise. So our goal was to map everything, all of the reptiles in the world. Turtles and crocodilians, pretty easy. They're big. There aren't very many of them. They're pretty well known. But for the squamates, which are the lizards, the snakes, and the amphisbaenians, the worm lizards, we just had no clue where a lot of them were. And so we had to use every data source available. So museum records, digital databases of museum records and other things like VertNet or GBIF or things like that. Any published source, basically any way that we could figure out where species were, that's what we started with. And at the very beginning, because we've never done this before, what we did was just the best, sort of the best resolution we could get. So in some places that's dots on a map. We know you can go to an area and people have gone everywhere, scientists have gone everywhere. And so we could put dots on a map. But I was in charge of Peru and Bolivia, 2 pretty big countries where I can't go every little single place in those countries. So what I had to do was I used atlas sampling, where we just divided the world into 1 degree longitude, latitude grid cells and presence or absence in those grid cells. And then also we predicted where else they probably were based on just our knowledge of the habitats and things like that. So we completed the first round of lizard maps in 2009, and then the snakes and amphisbaenians were added in 2012. And this was the first map that we published. So this is just a richness map. So it's not too exciting because it's just richness of all the species. You can't see what species is which. And also not a super surprising map. The Amazon, the rainforest of Southeast Asia, these are places that have a lot of diversity of reptiles. So it may not look like much, but this took years and years of a lot of people to get this together. And behind this are the individual maps of all of the species. So we published this in 2017, and you can go to our website, the GUARD Initiative, to see more details. So that was great, but we've got 44 publications so far. I'm not an author on all of those, unfortunately. But we're currently working on GUARD 2.0, where we want to do better than that. We're not satisfied with atlas maps. We want point localities. We want polygons for every single species in every place in the world. We have a web app, a Shiny app that's been created that will eventually be released for the public, where you can go in and type in any species and you can see the exact polygon distributions and also records and add data, maybe if you have it. We haven't gotten to the public part of this yet. But that's the idea. And we've also added, so as I said, we only concentrated on those parts of the world that didn't have hardly any knowledge, but now we said, okay, well, now we're trying to refine this. Let's go to places that we had some knowledge, but not very good. So we've added a lot more countries, more experts from different countries to try to complete this. And it was, especially when we were first setting it up, it's our first meeting, we sat down and say, okay, let's divide up the world. I'm in charge of this country. You take that one, you take that one. And we literally sat down like conquerors and decided who was getting each country. And there were some fights over people that wanted a certain country and also some countries that nobody wanted, like you take North Korea. No, you take, it's not like we can just go there. And it's, so there was some of that, but now we've got, I don't know even what the number is, over 40 people still working on this, trying to get the GUARD 2.0. We're trying to get that released in March. We'll see if that happens, but that's what we're working on. And so just some things that I found. So I was in charge of Peru and Bolivia, and they're I'm mostly just going to talk about the squamates because lizards and turtles and crocodilians are easy. But in just those two countries, there are 703 species of squamates, which is 7% of the squamates of the world. So these two little countries actually are add a lot. And just to orient you where we are, so this is South America, this is Western South America, and Peru and Bolivia here. And you can see major feature, obviously, is the Amazon basin, but also the Andes Mountains run through both countries. In Peru and neighboring countries, there is the Atacama, Sochura, Peruvian desert, whatever you'd like to call it, driest desert in the world. And then down here, you have some savanna habitats as well. So there's a lot of diversity of habitats in these areas, which has led to the diversity of reptiles and other species. This map is really ugly, but it's a way to display what I was dealing with. So I mapped all of the lizards and snakes and amphisbaenians in Peru. And in the end, I found that there were 15 1-degree latitude longitude grid cells with no reptiles ever recorded, which is crazy. Okay, right? So here's the Amazon basin. These red boxes are the areas where there are no reptiles recorded. I mean, do you really think that there are parts of the Amazon basin with no reptiles? No, that's crazy. And so the numbers are actually the number of lizards in each area. The red boxes are there are no reptiles at all recorded. And so this shocked me, it really shocked me. I just couldn't believe that that was the case. So why? I believe 100% that every single one of those grid cells, regardless of where it is, does have reptiles in it. It's just no scientist has recorded them. Of course, the local people know they're there, but a scientist has never recorded them. So if we just have greater sampling in these regions, then we'll get some species there. In Bolivia, it was even worse. Bolivia is a smaller country, but it had 21 grid cells that had no reptiles recorded in it. And that was not, the difference was not surprising to me because Bolivia has even fewer biologists working in it. But the reasons for these empty areas are a little bit different. Part is poor sampling for sure. But the other part is these very, very high elevations in the western half of the country, the Altiplano area. And there are two genera, one lizard genus and one snake genus, that are the only species that live in those areas. And then some of the Altiplano is over 6,500 meters in elevation. You're not going to get reptiles there. And there are parts of the Oruro and Potosi departments, the political states in Western Bolivia, that the entire thing is over 4,000 meters. So it's probable there really just aren't reptiles there. And this is a cool map. It's weird because of the orientation, so that's north. But I like this map because you can really see the elevational difference here, and that this whole side is really, really high in elevation. And so we probably just don't have reptiles in most of those places. But there are some empty cells in the Amazon and in the savanna areas too. So I'm sure that there are reptiles there. So I wanted to fix this problem. And so I wrote a grant, an NSF grant proposal for the pursuit program, the poorly sampled and unknown taxa, to go to the 15 grid cells with no reptiles and find the reptiles. And so it's meant to be a 4-year project. And so I did a little sort of pilot study, proof of concept, where I went to this one that had none in it. And I went there and I said, okay, I'm just gonna go. I had a very tiny grant. I took one student, we went for 6 days, and we just, tried to find reptiles. We found 11 species, which is certainly not all the species that are there, but it shows that there are some there and that they're quite easy to find. And this cell is right next to a pretty big city in the rainforest, Puerto Maldonado. So it was very easy for me to get there. I'm shocked that no one had ever gone to sample there before. So it's kind of a proof of concept that this is something that could be done. So far, the grant has been denied. So I have not gotten funding for this, but maybe one day I will be doing this project and filling in these numbers. Okay, so that's all I'm going to say about that, because that's not the main purpose of my talk here today. The main purpose is to talk about lizard malaria. And when I say lizard malaria, the first thing that everyone says, which includes today, is lizards get malaria. Yes, in fact, they get a lot of malaria. But if we think about human malaria, it's one of the deadliest diseases on the planet. And we Americans don't tend to think about that because we're here and there's not malaria here anymore. Well, there used to be malaria here in Florida. But Plasmodium falciparum killed 627,000 people mostly in Africa, but also some in Southeast Asia and other areas in 2020. So this is a major, major human disease that the normal number is about 500,000 deaths of people per year. It was higher that year because of COVID and less hospital access. But that's the latest data that I have. And malaria is caused by this protozoan called Plasmodium. So here are human red blood cells. Here are some more human red blood cells, and they have these little things in them. And those are the parasites. And these are white blood cells, so that's not what we're talking about here. But you can see all of these little parasites inside the red blood cells. But the thing that people don't realize is malaria first evolved in lizards, as far as we can tell. So humans can get 4 species of malaria, lizards can get 100, or slightly over 100 actually. And the current knowledge, as far as we can tell, malaria first evolved in lizards, then was spread to the reptiles, birds, mammals. And so there are a lot more malaria species found in lizards than any of the other taxa. But even though that's true, almost nobody studies lizard malaria. And I really started this mostly by accident, something I was always interested in, never really intended to study. But I had a student who was really interested in it. He said, let's do this. I don't even know how to do this. I don't even know what you do. But we learned together, another student, 2 students and I learned how to do this stuff. So the other important piece of this is that the state of Florida is just insane as far as invasive species go, as I'm sure all of you know. So as far as reptiles, over 40% of the reptile fauna here in Florida is invasive, did not come from here. In fact, every time I teach herpetology, I have to add more species to the list because there's a new species got established that came from somewhere and now it's living a great life in Florida. So let's think of why Florida is such a mecca for these endangered or for these invasive species. Some of the things are the extrinsic to the lizard. So Florida is just a great place to live because the subtropical habitats we have here are pretty similar to the tropical habitats where most of these species come from. So it works. But then if we think about the species that actually become invasive, they establish and become invasive, there are a lot of intrinsic features about them, like they can tolerate urbanization really well. They usually are generalists, and they in some way have superior competitive ability. So all of these things kind of add together to make them pretty good at invading new areas. So as far as the lizards in Florida, in most of Florida, we have two species of anoles, which I'm sure you're familiar with, the green ones, which are the native ones, and the brown ones, which are invasive. In most of Florida, that's what we've got. So let's go back in time though, back to the early 1800s. So here you see Florida. And back then, there was only a single species of anole in Florida. There was the green anole, Anoles carolinensis. And we can think of them as like the kings of Florida because you could use any microhabitat they wanted. In fact, they had really wide microhabitat usage, much more than this species does in other places where there are other competitors. So it probably had an unnaturally wide range of microhabitats, but it could do anything it wanted because it didn't have any competitors or no other lizards or other species that are very good competitors with anoles. But then in 1887, brown anoles were introduced, and they were introduced again and again and again, multiple times through ornamentals, plants, or various ways on ships, but not just in one place, but in multiple places. There have been lots of genetic studies to show there have been multiple introductions. And when they came, they were just better at kind of everything. So they took over most of the microhabitats, especially the ones associated with human dwellings, but also most of the areas they win. And they have now sort of pushed the green anoles out. Now, the green anoles aren't going extinct. They still exist. They've just been pushed off into marginal habitats. And I say marginal in quotes because it's usually the more natural habitats is where you find the green anoles, but marginal for the brown anoles. And so they've really taken over most of the green anoles' former habitat. So one thing I was thinking was that why are they such good competitors? All of those features that I listed before, but maybe another reason could be their susceptibility to disease. So maybe the brown anoles and green anoles, maybe they have the same susceptibility, or maybe they don't. And that's a question that we wanted to ask. So to speak more specifically about lizard malaria, there's 101 species that have been described so far, poorly studied, as I mentioned. Many things about it have been poorly studied, but in particular, things about their health and fitness, that what it does to the lizards is poorly studied, particularly here in Florida. So some lizard malarias that occur in California that do not occur here make the lizards so sick that they die. Doesn't seem like they do that here, but we're not really sure. So one of the things also just to point out, because this is relevant for what I'll talk about in a minute, these are lizard red blood cells. And you can see they have nuclei, unlike are the human red blood cells. Mammals don't have nuclei, but that makes these cells very robust and long-lived as opposed to the mammal red blood cells. But you can see that psychometacyte parasite right there. All these little things, these are all the parasites, the plasmodium inside the red blood cells. And we won't go into this in detail, but this is the life cycle of malaria in lizards. It's similar to the life cycle in humans. But the point of me showing this is there are many different stages that have to happen in the lizard and many other stages that have to happen in the vector in order for this to be transmitted, which is typical for many of these protozoan parasites. So there's only one species of lizard malaria in Florida, and that's Plasmodium floridense. It infects 29 lizard species in the range where it lives. So that's Central America, Caribbean, Florida, and Georgia. But in Florida, it only infects, as far as we know, 3 species. So the 2 anoles that I just mentioned, plus eastern fence lizards. I suspected it infects probably more than that, but that's all that's been tested. One thing that had never been studied is what is the differential infection prevalence of of malaria in these lizard species. So what I wanted to do for this very first study that I did with these two students was to see if infection prevalence is the same in the two species of anoles or different. And also I tested some other factors that could be related to infection just to see, because no one had done this before. So it's pretty exploratory, but really we wanted to know, is there a difference in infection prevalence? And then also to try to determine if the parasite actually hurts them, because it's not clearly known and we still don't actually know that. So these are the two students that were working with me. Field sampling is very simple. Go out, catch a lizard by hand or with a lasso. The student was really good with the lasso. Then we take a clip of their toe to get blood and smear that onto a slide, and then fix it and stain it. One thing that's important to point out is this species of malaria only infects red blood cells, not white blood cells, like some other malarias. And that's relevant to some of the analyses that we do. So we would, it's actually a pretty simple protocol. Anyone could do it. So you sit with a microscope and oil immersion lens, and you search through the red blood cells for 5 minutes. And if you find one, then that's a positive. And if you don't, that's a negative. I haven't done, but other people have done some PCR studies to see if that's a really good way to, if that works. And yes, it does. There's not a lot of false positives or false negatives. We were also looking at the parasitemia. So how infected is the lizard? Does it have very little infection, or are all of its red blood cells filled with parasites? And then we also wanted to know the sexual versus asexual parasite stages. So this is sort of thinking back to that life cycle I showed you. So here you can see the two stages of the parasite. And this is one I showed you before. So this guy right here is called a gametocyte. Gametocyte is the sexual stage of the parasite. The marant over there is the asexual stage of the parasite. Why do we care? Because if a vector, probably a mosquito, bites a lizard that has only marants, that mosquito will suck up that blood, but that cannot continue the life cycle, and therefore that mosquito will not be able to infect another lizard. However, if they get lizards that have gametocytes, then it can go on and continue its life cycle and be infectious to other lizards. Okay, and so these are some of the students doing that. All right. So for the first study, we looked at 3 sites in Central Florida. It was very important to us to work at sites where the lizards live syntopically. So sometimes on the same tree, you get the green and brown anoles, which is pretty uncommon in Florida these days, but we wanted to make sure that all the other factors, like the habitat factors, were constant. We had 101 anoles where we got good blood from. And here you can see just the infection prevalence. Brown ones, 11% of them, and green ones, 31%. Okay, right there, wow, it's almost 3 times as much for the brown or for the green anoles. And that was a significant difference. So lizards that were infected had between 1 and 24 cells infected per 1,000 erythrocytes. And so we looked at how many had marants and gametocytes. We also counted their red blood cell to white blood cell ratios. So white blood cells, as you probably know, are elevated when an animal is suffering some kind of infection. So this would give us a kind of indirect way to say, okay, is this making them sick? You know, are they actually responding to this? And we found that the species didn't differ with their red blood cell to white blood cell ratios, but if they were infected, they did. So it didn't matter what species it was, but either species, if they're infected, they had elevated white blood cells, which you would expect, but also we weren't really sure. We found a big seasonality infection prevalence. As you get later in the year, we had a lot more infected lizards. And we had this big switch from mostly marants, the asexual non-infectious stage, to gametocytes, the infectious stage, the sexual stage. And this is kind of a funny graph, but it's cumulative. So you can see that at the beginning, we didn't even catch our first gametocyte until well into the project. And then nearly all of them were gametocytes after that. So definitely a seasonal thing going on with the parasites, with the stages, life stages of the parasite. We were looking at these other factors. So that seasonal thing I mentioned already, we didn't find any patterns with sex, so males and females equally infected. We also didn't find any patterns with tail breakage. So these lizards can autotomize their tails and then their tails grow back. We had some thoughts that, okay, well, maybe if the lizard is sick, then they're more likely to get their tails broken because they can't run away. But the other side of that is if the lizard's sick, maybe it just gets eaten and dies and doesn't get its tail broken. So if those two things could have canceled each other out. I can't really make any, we found nothing, but there's no way to tell without doing a specific study on them. We did find that increasing lizard size was associated with increasing prevalence. And so this could indicate a couple of different things, non-exclusive, non-mutually exclusive things. Some people have suggested that malaria infection in lizards is actually chronic, that they just keep it. They don't clear it throughout their lives. So bigger lizards, older lizards are more likely to be infected because they were infected at some point during their lives. That's possible and maybe probable. Also, another possibility, bigger lizards have more skin, and so maybe they're getting bit by mosquitoes more often just because they're a bigger target. So one or both of those may or may not be true, but not something I've tested. So overall infection prevalence of both species, 16.8%, which sounds kind of high, but compared to infection prevalence of California lizards, it's quite low. And as I said, they have elevated white blood cell counts. And then we saw that the infection prevalence of the green anoles was much, much, much higher than the brown anoles. So this got us thinking, maybe this is another one of those things that the brown anoles have over the green anoles. They not only are they superior competitors, they don't mind urbanization, all of these things. Also, they don't get infected with malaria as much for whatever reason. There have been previous studies in the Caribbean that showed that malaria could mediate competition between species. These are different species of anoles and different malarias, but still, so that's not a far-fetched idea. So maybe there's a synergistic thing going on with the wider niche breadth that the brown anoles have, the higher tolerance of urbanization, and the lower rate of Plasmodium infection all came together to make them be able to invade Florida and also be superior competitors to this day. So as I mentioned, it's been very poorly studied. So we needed a lot more studies to try to figure out, is this one of the reasons that brown anoles were able to invade, and then do the lizards get sick? So we continued from there. And we did another study where we looked at the white blood cell and red blood cell ratios, which I already mentioned, but we said, okay, let's look at not just any old white blood cell, let's look at specific types of white blood cells. So we have a variety of white blood cells, as do lizards. And these are actually human one here, but lizards have similar ones. And the nice thing is microscopically, they're really easy to identify because they're such strange nuclei. These are weird shaped nuclei. So you can see an eosinophil is very different from a lymphocyte or whatever. So we thought eosinophils are known to be elevated when there's parasitic infection, like a protozoan. So we thought, let's look at the eosinophils and see if there's a difference. Another thing is in humans, we would call it neutrophil to lymphocyte ratios. Lizards have a homologous cell called heterophil. So heterophil lymphocyte ratios should be elevated when the animal is sick or under stress. So we said, okay, let's look at HL ratios. And let's look at eosinophils and let's see if we can get more idea about the being sick. And so one of my students was a lead author. And we use the same methods that I already mentioned, but we not only sampled in Central Florida, we also sampled in Southwest Florida. And in the end, we didn't actually find anything interesting about the eosinophils or HL ratios. As we had with the previous study, we did find there's the white blood cells were elevated in infected lizards in Central Florida. We had already seen that in the previous study. But in Southwest Florida, that wasn't the case. And even though we had a greater sample size in Southwest Florida. Like, well, that's weird. So they get infected just as much, but they don't seem to be as sick, maybe, as at least immunologically. And so I don't have an answer for why this is, but the best thing I came up with is that in Central Florida, the anoles are under constant competition. The brown anoles are under constant competition with the green anoles. In Southwest Florida, we didn't even study green anoles because they're so uncommon that we wouldn't have had enough sample size. So the brown anoles have been kind of released from competition, at least with the green anoles in Southwest Florida. And so maybe because of that, they're not under as much stress, and so they're not getting sick. This is pure speculation. I haven't tested it, but that's the best thing I could come up with for seeing the difference. The most recent part this paper is now in review is going a slightly different direction, but one of the things that anoles are famous for is having the dewlap, which is it's this extensible flap under the throat. And in brown anoles, it has a yellow margin and then sort of orangey red in the middle. Okay. And so males use this to signal each other. They are using it to signal other males and for territorial reasons. And they're also using it to signal females to try to impress them for mating. So both intra and intersexual selection happening. Another thing to note is that the vision of lizards is pretty similar to human vision, unlike a lot of species that have different vision. Lizard vision is very similar to human vision. They can see a little bit into the ultraviolet, but other than that, it's the same as ours, which is convenient when you're studying them because I'm seeing what they're seeing. So it's nice. If we were working on mammals or something, it would be harder. So some studies in the past have shown that dewlaps act as honest signals in territorial interactions. So that the male is signaling how good he is at fighting or whatever it is, and that has turned out to be an honest signal. Other studies have shown that females do select males with redder dewlaps. And other studies have shown that dewlap coloration signals quality, health, immune status. So I thought, okay, there's this hypothesis called the Hamilton-Zuck hypothesis, which states that secondary sexual characteristics, usually of males of the competing sex, are altered or made negatively affected by parasitism. So I thought, okay, well, anoles have this very important sexual characteristic. So maybe this is being affected by the parasitism. The Hamilton-Zuck hypothesis had two predictions. The first one was the interspecific prediction that in species that suffer from parasitism a lot, they would have more ornamental traits, so bright colors or feathers or whatever. And most of what's talked about was with birds, at least initially. But there's also the intraspecific prediction, which says that individuals that are parasitized will have reduced secondary sexual characteristics. So if we're talking about color, duller colors. So here is just an example. We have these nice cardinals. And so the female there, the male is that colorful red. And this guy are both colorful, but this guy is a lot brighter than that guy. So it's possible that this guy is not suffering from parasitism and that guy is, or at least that's what the Hamilton-Zuck hypothesis would say. So it's rarely been tested in lizards, and so I wanted to test it with the dewlaps of brown anoles. So my hypothesis for this section is that male brown anoles will have duller dewlaps if they're infected with Plasmodium floridensii. So we did all the same kind of field sampling for the other projects, but we use this handheld spectrophotometer we could pull out the dewlap and then get readings on the dewlap. So we would take 3 readings, 2 on one side and 1 on the other. We did the same microscopic protocol. And then we use this formula for quantifying relative brightness, which is known as luma, that would give us an idea of the relative brightness of the dewlap. We also just looked directly at the red color value, so of the red, green, blue that you get a lot of times when you use a spectrophotometer. We looked at red chroma values as well. And so we're testing differences in the average luma and average chroma per lizard to see if there's a difference in infected lizards versus not infected lizards. We also tested size because I had mentioned before that size could be important in infection. We didn't know if size could also be important in color of the dewlap, so we wanted to test that. And then we wanted to, so that's with infection status and with color. So we did all of those tests. So for this part, we had 97 anoles, all males, because that's all we were looking at. Various sizes, any male was subadults or adults, as long as there was a dewlap, we considered that lizard. And we didn't find any difference in size with infection status at all. And we also found, as far as size, that average luma and average chroma, red chroma, were not correlated to snout vein length. So the size of the lizard. So size was not important in this particular case. But the things we were actually most interested in. So here you can see there's a lot of overlap. What we're looking at here is infected lizards are in the black, and then orange is the uninfected. And it's really hard to see. Can't really see at all. Okay, but there's a line in there. So what you can kind of see on this, but you can see better in the numbers, is that the lizards that are infected have a much lower luma than on average than the lizards that are non-infected. Okay, incredible variation in the infected lizards, or sorry, in the uninfected lizards, but you can see that there is a difference, which was a very slightly but significant difference. Also, for the red, the red was a duller color also. I didn't put the numbers up here, but so infected lizards have brighter dewlaps and less red dewlaps. So think about that for a second, and then I'll explain why. So I was confused at first, as most people are, because the way we tend to use the word bright is not the way someone who studies optics uses the word bright. So bright, to like as far as light goes, is more white. Okay, when I say, if you say, oh, look, there's a pretty bird and it's brightly colored, that actually doesn't make sense, because brightly means less colored to an optics person, okay? And there's actually I've written a whole part of my discussion in this thing about how people in the past have interpreted the Hamilton-Zuck hypothesis incorrectly because they use the terms wrong with their spectrophotometer results. Anyway, so more white. So the lizards that are infected have less color and less red color, which goes together, of course. So that would tend to support the Hamilton-Zuck hypothesis, because the infection somehow is related to the less colored lizards. So there have been some studies in the past that have shown that European green lizards had duller throat and body colors if they're infected with some kind of parasite. This is a blood parasite. There are anoles we're looking at with ectoparasites like mites, found that as well. But then there have been studies that have shown no significant differences, studies that have shown the opposite, that the ones that were infected were more brightly colored. So for lizards, there's been evidence in all directions. This one particular study, though, that found brighter colors with infected lizards proposed that in this species, which are whiptail lizards, the brighter colored ones are much more aggressive, and so they're out running around instead of hiding in their burrows. So they probably had more chances to be bit by mosquitoes. So this is the first time anyone's tested the Hamilton-Zuck hypothesis with anole dewlaps and with malaria. And so we found support for the Hamilton-Zuck hypothesis. And parasites can devote more resources to the immune response. So if we're talking about carotenoids, which is one of the two pigments that makes up the color and the dewlap, carotenoids are environmentally derived. They can't make them. So and they're also similar, I don't know all the biochemistry of it, but similar things are used in the immune response. So if they are having an immune response, they have less energy to devote to taking those carotenoids and incorporating them into the dewlaps. But we really need more research to show why this is happening and how. This is really the first time of doing anything like this. The other thing that I would love to do, but have not gotten funding to do yet, is, yeah, okay, in most of Florida, we have 2 anole species, but you go to Miami, and it's just a playground of anoles. All of these species are invasive in Miami. No one's ever tested them for malaria. I did this little tiny study in Costa Rica where no one had ever tested, and we tested 6 species of anoles, found 5 of the species were infected with malaria. So I bet if I go to Miami and test all of these guys, probably most, if not all of them, will also be able to be infected, and be really cool to do some more things with that. But okay, I wanted to end by talking about what I'm doing with Archbold and what our new project is. So this is just getting going. It hasn't really we haven't started the field work yet. But we received a startup grant, Strategic Environmental Research and Development Program grant from the Department of Defense to work on Avon Park Air Force Range, which, as most of you probably know, is here in Polk and Highlands County. It's a huge area, 106,000 acres. And so what the purpose, the solicitation that we applied for SERDA was to look at synergistic effects of multiple stressors on threatened and endangered species. That was what the sort of solicitation was. So we're working in pine flatwoods, dry prairie, and Florida scrub, and some ecotones. And the factors that we're testing are fire frequency, fire ants, and grazing. And so what we're trying to do, and what the purpose of the solicitation is, to see not just how fire ants affect them badly, not just how fire affects them in various ways depending on the species, or grazing, is to see how these things are interacting with each other. And so that's the point. And so Ree Bowman is one of the co-PIs on this project, as is Angela Tringali. So for me, I'm doing the herbs. And so these are the species we're potentially dealing with. We don't even know necessarily that all of these species occur at that site, but these are all the species that could be there that are threatened and endangered. So indigo snake, sand skink, blue-tailed mole skink, Florida pine snake, short-tailed snake, gopher tortoise, and gopher frog. And then also many bird species and small mammals that our collaborators are going to be working on. So the methods-wise, we will be setting up 60 plots that will be arranged in the different habitats, areas of different fire frequency, and half of those will have the fire ants removed. And so my methods, I'll be doing drift fences with funnel traps and pitfall traps in some of the areas, and drift fences with funnel traps and camera trap arrays in other areas, because I'm not able to dig and put these traps in a lot of the bombing range, because it's a bombing range and they don't want to be digging in the ground and finding things I shouldn't find. So our first year's survey year, which is this year, hopefully we're still waiting on the funding to actually appear. It's been allocated, but we don't have it yet. So we will be setting up our areas, and we're just doing the survey. So we're seeing, based on a different burn history, we're going to see what's there and what fire ants are there, and so we can sort of make plans for the remaining years. In the remaining years, we will be doing a fully crossed experiment with habitat, fire frequency, and fire ant. And fire ants will be removed from half the plots. And basically, we'll see what happens to the populations of the species, populations and demography of the species. And as I said, we're trying to really determine the synergistic effects of all those factors. I didn't put grazing on here because we're also, our collaborators are also working on grazing, but we're not doing grazing for the herpetophone part. So the idea is to eventually provide management recommendations for, should they go spend the effort to kill the fire ants? What should they do with the burning? Those kinds of things, to be able to provide the military with recommendations of what to do with the areas to manage for the threatened and endangered species that they have. And hopefully we can safeguard or maybe even increase their populations on the range and other areas in the Southeast with similar habitats. Okay, so those are tons of students that are all co-authors with me in all of these papers, and I'd be happy to take any questions. Yes.
So, do you see any effect of plasmodium on hemoglobin marker in the
I have not studied that. Some people have looked at that. Yes, it depends on the species of lizard and the species of plasmodium. No one's looked at that in brown anoles, so I can't say for that. I mean, no, I can't really think of anything. I've tried to think of things, but no, I mean, nowadays they're in more natural habitats and less urbanized habitats, because that's where the brown anoles really take over. But we made sure to do the studies in areas where both of them are living in side by side. So that factor should be out. But no, I don't. No, I mean, I would think if I were going to guess before, that the brown anoles would be more infected because they're much more aggressive. They're running around much more. The green anoles are just sort of chilling. Tend to hide out more. But no, I don't. If you have any ideas, let me know. Yes. Okay, yeah, sorry. So for my Zoom people, so the question was, the first question was asking if we had studied hematocrit or something like that, which we haven't. And the second question was, do the green and brown anoles, are they ecologically different? That would explain this. And I can't think of anything. So that's something I didn't mention. So yes. So do the anoles have behavioral differences, and are the mosquitoes different? So it is true that in areas where there's sympatric, a lot of times the green anoles will be higher up in the trees. That's not always the case, but that is often the case. So here's the problem. We don't know what the vector is. There have been hypotheses, and then I just last year happened on a paper with a new hypothesis. It's probably a Culex mosquito, but we don't know which one, and we're not sure. So that's a big problem. Yeah, they've done experimental infections, experimental mosquito infections, I guess, and a lot of mosquitoes can carry it, but whether they're actually doing the whole life cycle in it, probably not. So we don't know. So that could be something, but yeah, but we don't know. I need mosquito people to help me out on this one. Angela.
I'm glad that you mentioned the experimental infections, because I'm really curious if there's any plans change the advantage.
Yeah, so the question was if there are experimental like treatments for malaria for the lizards that we could somehow I don't know, level it out, and then that would be so cool. I've never actually thought of that in particular. But I mean, we know nothing about lizard malaria, so I don't know how you find a treatment for lizard malaria when we don't even know much about it, but that sounds like a really cool idea. Yeah.
Yeah, I'm curious about how. Yeah. Brown anoles favored by
Yeah, so the question is if spraying for mosquitoes could somehow affect the dynamics of this, and that's also really cool. I hadn't really thought of that before either, if that could be an equalizer potentially. So I have lots of other little projects that I've worked on, and one that didn't work very well because I had students that just didn't try very hard, but I will go back eventually. But one very cool one that I really do think could work is what we did is we went to the University of Florida Museum and got specimens from all the way of green anoles all the way back from the 1800s. And I was able to get blood, and actually I can see plasmodium in the blood from these specimens, which is so cool. But my students just did a really bad job. So I have to go back and look at all of the slides again, and I have not done that. So but that doesn't really answer your question, but that would sort of see like when before the brown anoles were here, were they still getting as much infection as they are now? That was kind of an indirect answer to your question, but yeah, that's really interesting to think about. I don't know how I would do that, but it'd be very cool.
I'm just thinking about the high densities of realms in the areas where they are. Maybe just that competition in those high density, interspecific competition is having negative effects on the immune function of the green anoles in that situation. Maybe this.
Yeah, so the question is if the areas where they're syntopic, that because of the competition, that's negatively affecting the immune function of the green anoles. So yeah, I think so, but why wouldn't it negatively affect the brown anoles as well? I mean, maybe they just have inferior immune systems. It could be.
I don't know a lot about immune function.
I mean, I don't know much more than what I've learned from malaria stuff, but.
Yeah, comparing them in lower density or less competitive situations.
Yeah, well, that's in in South Florida, in Southwest Florida. So in the Sarasota area, we didn't even study the green anoles because they're just so uncommon, and that we didn't find that big difference in infected. So that's what I assume. I shouldn't assume, but what I guess maybe would be the reason. But yeah, I don't know. Angela?
I was thinking that would be
Yeah, so the question is maybe the brown anoles naturally occurs at higher densities. That's possible, but I don't think so, because before the brown anoles were here, the green anoles were just everywhere. I mean, even when I was a kid, I remember them being everywhere, as abundant as the brown anoles were up in North Florida before the brown anoles got there. But it could be, but yeah, I mean, everywhere else where it was such, as I tried to say with that little map, that it was such an unusual thing that in Florida there was only one native species, because everywhere else where you have anoles, you always have multiple species. So it's a really weird thing that allowed them to expand their niche way more than they should have, than they normally do in other places. Reed?
So looking birds is actually pretty. Right.
Yeah, so so the question was, in birds, there's a lot of connection between the stress and competition. So just I already forgot, and immune response. And so, has anyone done that kind of thing in lizards? They have, but not in looking at multiple species. So they've looked at how malaria, they've taken the cortisol and all these kind of things. And looked that malaria does negatively affect them, but I've never tried to do more than one species at a time in a place. So yeah, that's definitely something to do. Hillary?
maybe I missed it at the beginning, but the brown anoles in the Caribbean, how many malaria species have they been exposed to over their life?
They have, yeah, so it's not that they've never seen. Yeah, so so as far as we know, I mean, there are other species of malaria. Actually, Plasmodium floridense is in a lot of the Caribbean. So it wasn't that when they came here, it was the first time they'd experienced it. So they had, as far as we know, the species has always been in Florida and infected whoever was here at Florida and a lot of the Caribbean islands. So, and even I think a little bit into Central America. So it wasn't a new experience when they got here.
I'm just all.
Okay, yeah, so the question was where the brown anoles came from, maybe that they had, because they had been exposed to multiple species of Plasmodium, maybe they had a better immune system for that. It's possible. There are also green anoles in the Caribbean natively too, but not, that was never, the ones that were always in Florida had only ever encountered the one species. So yeah, possibly. But I think in most places where there are multiple species of Plasmodium in the New World anyway, they're usually one or two. It's not a big number. Yes.
Yeah, one question on Zoom.
Okay.
So that you're in the first session.
I don't remember. Yeah, I think it depends on which part you're in, but yeah, I think we got good blood from around 100 for the first study where we did green versus brown. There were more browns than greens, but we had a pretty good representation of the greens. In fact, one of our sites, we had a lot more greens than browns, which was very nice. Oh, there's another.
Yeah, I know that the dewlap, that there are various reasons for it, like attracting females. I noticed we have a lot of them. It seems like when I get close to them, they really start doing that, and not dramatic. But I was wondering, are they also trying to like scare them? Or is that just coincidence?
Yeah, so the question is, are the anoles seem to react to people, and so are they using it as a predator deterrent? So there are a lot of studies that have looked at this, and they've mostly found that doesn't seem to be the case, which I agree with you. Like I see them, they seem to be doing it to me often. But the studies that have actually tried to see if dewlaps are used for predator deterrence have found that have not found that. So I don't know. It seems, I mean, I'm with you. It seems like they like to react to people, but yeah, the studies have shown that that doesn't seem to be one of their functions. Maybe they just like you. Just say, hey.
All right, well, thanks so much.
Thank you.